Gray hair is commonly understood as pure genetics. Your parents went gray at 40, so will you. That's partly true. But research over the past decade has revealed something more interesting. The process of hair graying is significantly modifiable by biological inputs. Oxidative stress, nutritional status, chronic psychological stress, and even smoking all accelerate the timeline. And some of these factors are reversible, with documented cases of gray hairs returning to pigmentation after specific lifestyle changes. A remarkable 2021 study published in FASEB Journal by Balaka and colleagues documented individual hairs that turned gray during periods of high stress and then regained pigmentation after stress resolution, using proteomic analysis to verify the timeline. The lead researcher summarized, "These findings demonstrate that gray hair caused by psychological stress is not permanently fixed, and that under the right biological conditions, pigmentation can be restored." Stress doesn't just cause gray hair, it's doing so through immeasurable. Hair biology research has advanced significantly in the past decade, with the mechanisms of melanocyte function, oxidative stress, and stem cell depletion in hair follicles becoming well characterized. The science of graying has moved from its all genetics and inevitable to a more nuanced model that includes modifiable biological inputs. In this video, we are going to see exactly how graying happens at the cellular level, which factors accelerate it beyond your genetic timeline, and what the research shows can meaningfully slow. Hair gets its color from melanocytes, pigment-producing cells located in the hair follicle bulb. Each hair follicle contains a small reservoir of melanocyte stem cells that replenish active melanocytes with each hair growth cycle. Graying occurs when this stem cell reservoir depletes and fewer active melanocytes are available to pigment new hair growth. The rate of stem cell depletion is partially determined by genetics, but research shows it is significantly accelerated by oxidative stress, UV exposure, and chronic. Oxidative stress within the hair follicle is the most well-documented accelerant of graying beyond genetics. Research by Trueb showed that melanocytes are particularly vulnerable to reactive oxygen species. They have naturally lower antioxidant defenses than surrounding cells. Hydrogen peroxide, a byproduct of normal metabolic activity in the follicle, accumulates with age, and when antioxidant systems are overwhelmed, it bleaches melanin in the hair shaft directly. The Balaka-FASEB study that documented reversible stress-induced graying found that the mechanism involves the sympathetic nervous system's norepinephrine release during acute stress. Excess norepinephrine causes melanocyte stem cells in the hair follicle to rapidly divide and differentiate, exhausting the stem cell pool that would otherwise regenerate pigment-producing melanocytes across years. A single acute stress event, if intense enough, can deplete the follicle's stem cell reserve years ahead of its genetic schedule. Tobin's comprehensive review of melanocyte biology in hair follicles established the current model of graying. Melanocyte stem cells sit in the hair follicle bulge region between growth cycles. With each cycle, some stem cells differentiate into active melanocytes that migrate to the bulb and produce pigment. Over time, each differentiation event reduces the stem cell pool. The rate of depletion determines when a follicle runs out of melanocyte progenitors, at which point the hair grows without pigment. Vitamin B12 deficiency is one of the most consistently documented nutritional causes of premature hair graying. Multiple case studies and cohort analyses have found that B12 deficient individuals have significantly higher rates of premature graying, and that graying has reversed in documented cases following B12 supplementation. The mechanism involves B12's role in protecting DNA from oxidative damage and its requirement for the methylation cycle that maintains melanocyte function. Research on copper and hair pigmentation confirms copper as a required cofactor for tyrosinase, the enzyme that catalyzes melanin synthesis. Without adequate copper, tyrosinase activity is impaired, melanin production drops, and graying accelerates. Populations with copper-deficient diets show higher rates of premature depigmentation. The key dietary sources of copper include liver, shellfish, especially oysters, dark chocolate, nuts, and seeds. The Power Science Advances 2021 paper established hair as a novel biomarker of stress and biological age. By analyzing individual hair strands proteomically, mapping the proteins deposited in the hair shaft over time, researchers could reconstruct a precise timeline of when stress events occurred and correlate them with the onset of depigmentation. This methodology confirmed that psychological stress rapidly depletes melanocyte progenitors, and that relaxation following stress can allow partial restoration of pigmentation in follicles where some stem. The anti-graying protocol that the research supports focuses on 3 targets: nutritional adequacy, oxidative stress reduction, and chronic stress management. For nutrition, ensure B12, folate, copper, and zinc adequacy, all with documented roles in melanocyte and melanin biology. 4. Oxidative stress, antioxidant-rich diet, smoking cessation, UV hair protection, and reducing dietary sugar and processed food. 4. Psychological stress. The evidence from the FASEB study specifically validates stress reduction as a hair pigmentation intervention, not just a general wellness. The realistic timeline for seeing any effect from anti-graying interventions reflects hair biology. Individual hairs grow about 0.5 inches per month, approximately 6 inches per year. Any biological change you make today will only become visible in the new hair growth that emerges from improved follicles weeks and months from now. The gray or pigmented shaft already grown will not change. Immediate actionable steps. If you smoke, the evidence on smoking and premature graying is among the strongest in the field. Stopping is the most impactful single intervention available. Get B12 tested, especially if plant-based, and supplement to above 500 picograms per milliliter if deficient. Add copper-rich foods. Liver once per week, a handful of pumpkin seeds or cashews daily, dark chocolate. If graying accelerated dramatically during a specific period of high stress, then the FASEB evidence suggests some reversibility is possible if the stress has been resolved. Give it 3 to 6 months with good nutritional support and stress management. If you notice gray hairs appearing well before age 30, then nutritional deficiency testing is particularly important. Premature graying before 30 in particular correlates strongly with B12 deficiency and should be investigated. Hair graying is a biological process with a genetic baseline that is meaningfully modifiable by lifestyle and nutritional inputs. Oxidative stress, B12 deficiency, copper insufficiency, smoking, UV damage, and chronic psychological stress all accelerate depletion of the melanocyte stem cell reserve that gives hair its color. Addressing these factors doesn't guarantee reversal, but the research clearly shows it can slow the process and, in some cases where stem cells remain, may allow partial restoration. Understanding the science behind what happens to your body, even your hair, gives you real information to work with. MedBrief Today is here to make that science clear and actionable. Keep watching to keep learning. If today's video helped clarify something, share it with someone who needs it. Most people never see research summarize this clearly. That's exactly what this channel is here to do.